My plan for the next half hour or so is to share our ongoing work on a new method that we developed called the polygenic transmission disequilibrium test, and to share what we've learned about autism spectrum disorders from our work in this area. A brief outline for the next half hour is to introduce some of the motivating questions in autism, to then talk about the development of the method, and then to share some of the findings in autism that we've come to with this method. So autism is a very heterogeneous diagnosis characterized by impairments in social communication and repetitive and restrictive behaviors. And one of the areas where there's great heterogeneity in phenotype is in cognition, where particularly over the last few decades, there's been a real diagnostic expansion at the high functioning end, shown by the increasing ASD prevalence rates and the decreasing rates of mean intellectual disability, showing that there's been this expansion of the high functioning end characterized by IQ roughly at the general population mean. Along with heterogeneity in phenotype, there's also great genetic heterogeneity. At a population level, most of the genetic liability for autism resides in common inherited polygenic variation. There is also a role for rare inherited variation and de novo variation. And importantly, while on an individual level, de novo variants can be a very strong effect, for instance, having a very strong association with IQ, at a population level, ASD as a whole, most of the liability resides in common polygenic variation, and that's where we focused in on our method. There are a number of existing techniques for associating classes of common variation, such as LD score correlation, and that has produced many important results in the common variance space. In the case of autism, there have been some counterintuitive findings. For instance, here, I'm pointing to a positive genetic correlation between autism and educational attainment, i.e. IQ, as they have a very strong genetic correlation, educational attainment and IQ. And this is puzzling because on average, individuals with autism have lower IQ. So the question that's been in the field for a while is whether this finding was the result of, for instance, case ascertainment biases. If, for instance, individuals ascertained for autism cohorts tended to be of higher socioeconomic status, then you would see the spurious association in this case. So one of the advantages that we were looking for in our method was a way to deconvolute these potentially confounded findings, such as this one. The tool that we use to get at common variation in our method is called the polygenic risk score, and I just briefly wanted to go over how those are derived, since I think that's important to understand for our method. So very briefly, a polygenic risk score is just a quantitative estimate of genome-wide common variant predisposition or risk for any measurable phenotype. And these polygenic risk scores are derived from the genome-wide association studies for that trait. So if you do a genome-wide association study for autism or for height, you get out an association between individual markers in the genome, or SNPs, and their association to the trait, such as schizophrenia or height. And when we look at the results of genome-wide association studies, we're most used to looking at the SNPs that reach Bonferroni corrected statistical significance, so 5 times 10 to the negative 8 typically. There's a lot of polygenic signal that resides below this threshold. So our polygenic risk scores typically include hundreds, thousands, or tens of thousands of SNPs below this threshold to create the maximally powered polygenic risk score. Just briefly, a tutorial on how these are calculated. So in the table at left, we have our SNP phenotype associations from the GWAS. So SNP RS1 with reference allele A is positively associated with height. Then we have our individual level genotype. So this is the cohort that we're interested in scoring. Maybe individual 1 at that SNP is homozygous for the reference allele AA. So then the contribution to the polygenic risk score from that SNP for that individual would just be 2 times the effect size of that SNP, so 2 times 0.3. And then you would sum up across the genome for that individual and then do that for all individuals in the cohort, and you would come out with some number, 0.4, 0.1. The number itself doesn't mean anything, but that just says that individual 1 is genetically predicted to be taller than individual 2. It's that framework that we go on and apply in our method. So if you continue with the example of height, imagine that you ascertained a cohort of trios, so offspring and parents, and you ascertain them at random. On average, offspring would be the sex standardized mean of their parents' height. And in polygenic risk score space, the mean polygenic risk score of the offspring would just be the average contribution from the parents. And if we plotted the distribution of the polygenic risk scores of the offspring and the parents, you wouldn't expect any average deviation between the average polygenic risk score, which here has reduced variance because it's the average of mom and dad, and the polygenic risk score distribution of the offspring. However, we know that offspring are not always the sex standardized after their parents, and if we were to ascertain a cohort of children who were significantly taller than their parents, we would expect an average upward shift of the polygenic risk distribution for height, shown here on the right as the PTDT deviation. Not particularly surprising, as you would expect that if you are ascertaining a cohort for tall kids, they would on average have higher polygenic risk for height. And it's this framework that we can take into any ascertained cohort, such as I'll show shortly in autism. So how do we quantify this? Well, on an individual family level, we calculate the PTDT deviation as the amount that the child's polygenic risk exceeds the average polygenic risk of the parents. And then we standardize by the standard deviation of the parents' polygenic risk distribution for 2 reasons. One, so we can express our mean PT deviation in terms of a meaningful value, in this case, in terms of standard deviations on the average parent distribution. And also, so we can compare PTDT deviations between different classes of polygenic risk. If you then plotted the distribution of PTDT deviations for a cohort, for instance, in the case of a cohort ascertained for being taller in the offspring than in the parents, you'd expect a slight upward shift of this PTD deviation, which we expect in this case. And this distribution has a mean and a variance that we can plot in a graph like this, and then we can simply perform a one sample t-test on that distribution against a null expectation of 0 to see if there is an average upward shift. And I think it's important to understand what this finding means. So what this is a clear association between the ascertained trait, whether it's a cohort ascertained for height in the offspring, whether it's cohort ascertained for autism in the kids but not in the parents, and an association between that trait and the class of polygenic risk in question. And importantly, as a family-based cohort method, we are broadly immune to biases by population stratification and case ascertainment biases. So to get back to the positive genetic correlation between autism and educational attainment from before, some people thought that this was because you were systematically ascertaining autism cases of, say, higher socioeconomic status, which would drive this spurious correlation. But because it's a family-based method here, you are broadly immune to these biases because the comparison is within the family. And some of you may recognize this as just a 21st century polygenic version of the TDT, which looked at single variants, but here we're just applying this to classes of polygenic risk. The cohorts that we calculated polygenic risk scores for, about 6,500 in total number. The first cohort was the Simon Simplex collection of about 2,500 families. Just over 2,000 are quads. So we have mom, dad, the autism case, and the unaffected sibling. Importantly, the parents are also unaffected, as well as nearly 500 trios, so without the unaffected sibling. All members in the Simons cohort were genotyped for common variants, and about 90% were exome sequenced, which allow us to get a de novo variation, which has been strongly associated with neurodevelopmental delay, and I'll talk about that a bit later. For our replication cohort, about 3,800 trios from the PGC autism group, and all members here were genotyped for common variants, but no exome sequencing included in our analysis. And what we find when performing PTDT on the Simons cohort, here the nearly 2,600 probands, is across autism polygenic risk in the left panel, schizophrenia polygenic risk in the center panel, and educational attainment polygenic risk in the right panel, that you see over-inheritance of these classes of polygenic risk, which we claim is evidence of clear association between these classes of polygenic risk and being a ascertained autism case. On the other hand, the unaffected siblings in the Simons inherit the expected amount, i.e. no systematic deviation between the average polygenic risk of the parents and the polygenic risk of the offspring. We were able to replicate the primary finding in the Simons with the remaining nearly 4,000 families in the PGC. And then we combined the Simons and the PGC cohorts for our downstream analyses, stratifying by sex and IQ. So again, what we see here is evidence for clear, unconfounded evidence for the association between these classes of polygenic risk and being a ascertained autism case. And not shown here is the negative control that we performed using BMI polygenic risk. So just showing that a class of polygenic risk that we didn't think would be associated with autism indeed appears null when performing PTDT in the Simons cohort and the PGC replication cohort. We then showed that this finding was independent of proband sex, so stratifying by male and female probands, also independent of proband IQ, which is an interesting finding, particularly in the right panel. So we see that autism probands with low IQ in the intellectual disability range are still genetically predicted to be the smartest members of their family. This holds when excluding cases with a strong acting de novo variant, as I will discuss, and it raises questions about whether IQ in the ID range and autism means the same thing as IQ in the ID range without autism. We were then interested using exome sequencing for the families to look at the role of de novo variation in autism, because there's been very consistent associations in the past between the role of de novo variation in autism and in neurodevelopmental disorders broadly. So we went to define a class of strong-acting de novo variants in the Simons collection. So we defined two classes. The first were de novo protein truncating variants, and we filtered by those that were not observed as standing variation in the exome aggregation consortium, and those that were affecting a gene highly intolerant of loss of function variation. The second class were de novo copy number deletions, and those that either affected a gene intolerant of loss of function variation or a deletion greater than 500 kb. And we combined these two classes of de novo variants, which we call CDNVs, and in the Simons, these were observed in about 1 in 10 probands. Indeed, these variants are strongly associated with autism in the Simons, seen in 9.4% of cases and 2.6% of controls, or the unaffected siblings, strongly associated with reductions in case IQ, and also strongly associated with adverse neurodevelopmental outcomes in the Simons. So the graph here is showing the association between contributing de novo rate in the Simons probands and the counts of neurodevelopmental outcomes that we see in the Simons probands. So this is count of delayed walking, seizures, and intellectual disability, and a strong positive association between the count of these events in the Simons and contributing de novo rate. So just to say, which is giving us confidence that we are looking at de novos that are strongly associated with adverse phenotypes. So our question was then, even for Simons probands who carry one of these strong acting de novo variants, which have been previously very consistently associated with autism, do you still see polygenic overinheritance, the role of common variation playing an etiologic role in autism? And for autism polygenic risk and schizophrenia polygenic risk, you still see evidence of this overinheritance, which we think has a number of interesting implications, showing for the first time the role of common rare variation contributing etiologically to autism. And also, given that these are de novo variants that broadly predispose to neurodevelopmental delay, the idea that a particular common variant load could steer one with these de novo variants towards a autism diagnosis as opposed to another neurodevelopmental diagnosis. What's nice about PTDT is that while we developed it in autism cohorts, it's applicable across any ascertained cohort and across any class of polygenic risk. So we are in progress working on classes of autoimmune polygenic risk, getting in questions of paternal age across cohorts in autism and schizophrenia. But it's really broadly applicable to any ascertained cohort across any phenotype. And it's really not a method that's very difficult to implement. So I just want to talk about cases when it would be potentially right for your analyses, and then a brief tour through an analytic tool that we've developed to make it a little easier to carry out these analyses. So if you want to associate a class of polygenic risks, such as educational attainment, with case status, what you need to do this are two things primarily. So you need a GWAS to exist of the class of polygenic risk that they're interested in, and you just need the summary statistics from that GWAS, so the SNP phenotype associations, and then a genotyped cohort for the ascertained trait that you're interested in. So a cohort of kids who are taller than their parents, or a cohort of cases with autism with unaffected parents. And there are a number of family structures that are compatible. The best are quads, because you not only have a affected case, unaffected parents, and as well a unaffected sibling, which allows a negative control. If the unaffected siblings don't exist, then trios work fine as well. And you can also use case unaffected sibling cohorts, assuming that the unaffected sibling is just a noisy proxy for the average polygenic risk of the parents. So not ideal, but if that's what you have, then that works too. We've developed a PTDT analysis tool to make it a little easier to perform these analyses, and what you feed into it is a polygenic risk score file. So on your own, through a program like Plink or elsewise, to calculate polygenic risk scores for your trait of interest for your cohorts. So say with the Simons, you use Plink to calculate polygenic risk for educational attainment, and then this is just a simple text file of the unique individual ID and their polygenic risk. And then a family structure file, which specifies for the program which individuals are in which families. So you just, so again, this is just a text file where the individual IDs from the polygenic risk score file appear in the family structure file, telling it which proband, which father and mother appear in which family. And then it gives you basic PTDT results, which is the mean and significance of your test as some basic QC. And it's compatible with quads and trios, and also allows you to easily subset your data on, say, families where the proband has a contributing de novo. So to wrap up, PTDT is a tool that leverages the benefits of family-based cohorts to perform genetic association in a way that is broadly immune to confounding bias by ascertainment and ancestry. And while we used 2 autism cohorts to develop the method, it's generalizable to any ascertained cohort across any class of polygenic risk. And there's a, and you just go on GitHub to grab that tool. When we applied it to autism, we used it to clearly associate classes of polygenic risk, including higher educational attainment, with ASD liability. So again, this is overcoming some of the potential confounders that we thought were behind this finding and others. And then we used the exome sequencing from the Simons to analyze together common and rare variation, and to show that polygenic risk contributes to ASD liability, even in cases with an extremely strong acting de novo mutation. Finally, a big thanks to everyone involved in this project, particularly Elise, Emily, and Mark here at MGH and the Stanley Center, and all our collaborators around the world. And